Hello students, welcome to Capman Academy. Today we are going to talk about molecular mechanism of Agrobacterium mediated gene transfer. As we already know that Agrobacterium is a gram-negative rod-shaped bacteria which infects the plants and causes their crown gall disease. And it contains bacterial chromosome and a specific plasmid which is known as Ti plasmid, which consists of tDNA and Vir regions, right? So today we are going to discuss about how this Agrobacterium tumefaciens causes pathogenicity in the host plants. So we are going to see its molecular mechanism. So basically it is a two component system, right? And it is based on different type of factors. So first factor here is the activation of chemotaxis system, right? And this chemotaxis system here act as a sensor for chemicals which are released by the wounded sites of host plant, right? So this wounded site actually, they make the bacterium to move to these sites, right? And this attraction of the bacterial to the wounded site is called chemotaxis. Another change which occurs as the chemotaxis system gets activated, chromosomal virulence genes system, for example, CHV, chromosomal virulence G, and chromosomal virulence I system gets activated, which confirms the acidity or sugar release from the wounded sites, right? And they leads to the activation of another system, which is Vir A and Vir G system. Vir A and Vir G system, they play a very key players during the pathogenicity because they promote the coding of Vir protein, which leads to the high expression of Vir genes in the Agrobacterium, right? And it occurs when chromosomal another virulence gene system E attaches to the monosaccharide which is released from the wounded site, which is released from again wounded site. So as you can see in this image, as the wound occurs in the plant cells, right? It actually through the chemotaxis attracts the Agrobacterium tumefaciens cells, and the molecules it releases, those are phenolics, sugars, and acidic, right? And during the release of these chemicals, they attract the bacterium and activate certain type of system there. So first is the activation of chromosomal virulence gene system, which I told you, G and I, which then activates the Vir A and Vir G system here. And this Vir A and G system then leads to the activation of Vir gene expression, right? So now we are going to discuss about, so this is the first step in which the Agrobacterium tumefaciens, it's getting attracted towards the wounded plant cells, right? So this signal cascade activity by acidity and phenol and sugar molecules, they are responsible for the activation of Virgin system, right? And then they leads to the another step, another important step of the infection, which is the signal receiving, right? So when the chemotaxis system activates the Vir A and Vir G genes, then they starts the another cascade activity, which is the signal receiving. In this case, first the signal comes to the protein which is known as with A protein, and it is a membrane receptor. It is a membrane receptor histidine kinase, right? And it contains four domains. It contains four domains. First domain is periplasmic domain, whose function is to percept the monosaccharide, to percept the sugar and slight acidity, right? Another domain is known as linker domain, and its function to recognize the phenolic compounds which are released from the wounded sites. Another domain is called kinase domain, and its function is to catalyze the phosphorylation, right? Another domain is known as receiver domain, and its function is to activate Vir-G protein, which act as a intercellular response regulator, right? And activates the expression of other Vir genes. So when this Vir-G gene gets activated, then the Vir-A protein, it gets autophosphorylase by the kinase domain. This by its own kinase domain, right? And it transfers its phosphate group to Vir-G protein, and it initiates the transcription of Vir operons, right? So as we can see in this image, what is happening here, that the signals are coming from the wounded site of the plants, and from there Vir-A, which is acting as a membrane receptor and contains multiple type of domains. One is periplasmic domain, second is your linker domain, third is your kinase domain, and four is your receiver domain. As the signal received to the receiver domain, it activates the Vir-G protein, and as the signal received by the two component system, which is Vir-A and G, then the next step starts, which is the Vir gene expression, and after that the tDNA production start, which is a very major step in the Agrobacterium tumefaciens pathogenicity, right? So during the tDNA production, a complex of protein is formed, which is called relaxosome complex, right? And in this protein complex, different type of Vir proteins, for example, Vir D2, D1, C1 and C2 proteins, all attaches to tDNA sequence, right? As we can see here, these are the tDNA, and it has left border and right border. So it actually, all these proteins, they attaches to the tDNA borders here, right? Like this way. And they produces the cuts in the tDNA at the border regions and leads to the linear single stranded tDNA production. This occurs due to the protein which is called VIR-D2 protein, because D2 here, it is a exonuclease enzyme. It acts as a exonuclease enzyme and it leads to the production of single-stranded tDNA production, right? And this complex formation between the VIR-D2 and tDNA is called D2 tDNA nucleoprotein complex, right? Further, this complex is being cleaved by another VIR protein which is called VIR-D1. So VIR-D1 here, it cleaves the VIR-D2 protein. It cleaves the VIR-D2 protein from the superhelical T border. From superhelical T border, right? If it is your tDNA, then here your D2 is attached and here it actually forms the superhelical structure, right? And this superhelical structure, it is cleaved by the VIR-D1 protein, which actually leads to the D2 protein cleavage from the tDNA strand, right? So here VIR-D1 is actually acting as a type 1 DNA polymerase. Sorry, topoisomerase. Topoisomerase, because as we know, the function of topoisomerase is to release the superhelical structures which are formed in the DNA, right? And C1 and C2 proteins which are also present here with the tDNA, C1 and C2 proteins, they act as tDNA production enhancer. So they actually enhance the tDNA production, right? So due to this, finally we get a single-stranded tDNA, right? And now this tDNA is then further gets recognized and transferred into the bacterial cell. So before that, another process needs to happen, that is the adhesion of bacterial cell. And the step is adhesion of bacterial cell with host cell, right? So this one occurs by the two types of binding system. First one here is your unipolar polysaccharide, which is also known as UPP, dependent polar attachment between bacterial cell and host cell, right? Another system for the adhesion is the T-pilus mediated attachment. So when this attachment occurs, host bacterium actually, it releases its single-stranded tDNA into the host. So this is how the tDNA gets transported into the host cell. by the formation of pilus. Now, during the adhesion, there are other Vir proteins which actually plays very important role, and that we are going to see in our next lecture. So that's all for the today. Thank you.